All right. Hey, Jennifer. So
Hi there. Great.
I'll
Let me
pass over
share.
to you. Just while you're sharing that, just to let everyone know that, yeah, Jennifer's from Southwestern University, the Kinesiology department. All right, where you go.
All right. All right. So everyone can see my slide now, hopefully. Awesome. Great. All right. So yes, I'm Jennifer Stokes. I'm an assistant professor in the Department of Kinesiology at Southwestern University, which is just outside of Austin, Texas. And I really appreciate this opportunity to share with you a LT lesson that Dee and I created on the endocrinology system. So the lesson itself follows a lot of what Dee described previously in terms of how it's scaffolded or structured. So we start with an endocrine system review, which really covers just some basic endocrine knowledge that the students would have already covered in class. And then it moves on to some application and practice of that knowledge as well. These particular activities are all low stakes and provide instant feedback for the students, which is great to assist with their learning and kind of relearning and reevaluating that endocrine system content, which is often very tricky. And then the students move on to review some experimental data and diagnose virtual rats. So this might be a very new thing for them, but also a great learning opportunity. In terms of administration of this lesson, it really has a lot of flexibility with it. I personally did it as a lab activity as part of my course, but I believe Dee did it as part of her lecture course. It can be set up as group or individual as well. I ended up doing kind of a hybrid situation because I implemented this lesson as the students had moved online in the spring due to new COVID restrictions. And so while it was an individual activity, I did encourage the students to work in groups. Which would have to essentially facilitate group work as well. And it can also be synchronous or asynchronous, really just depends on how you decide to set it up in your course. The learning objectives for this lesson include describing the effects of exogenous hormone administration on endocrine gland function, as well as understanding the role of negative feedback loops, which can be particularly tricky in the system and endocrine pathways, and then develop and defend a hypothesis based on scientific data. So again, depending on the level of your students and the content of your course, these could be some pretty challenging learning objectives, but all really good skills for an A&P course and an A&P student. The resources for this lesson include a paper that was published in Advances back in 1997. So you may already be familiar with this course if you teach the endocrine system in your class. And so we actually use the data from that paper to build our experiments for this lesson. And then we also use Dee's book, Human Physiology and Integrated Approach for the Review Activities. That's the book that I use in my course. And so the students are already familiar with the text and the figures. So it's a great way for them to review. And I'll show you that here in a minute. The activity itself actually has 4 different components. So we're starting again with that review activity with the goal of just an interactive review of material already covered in class, get the students on the right track to complete the lesson and the more challenging material coming up. And then we've got some application with endocrine predictions. And so here the students are filling in a table, making predictions about what would happen in the pathways, and I'll show you an example of that as well. And then we've got our experiments. So here students are going to be reviewing data that's presented to them, and then they're going to be able to draw some conclusions and more importantly, in my opinion, defend those conclusions and their hypotheses. And then we like to give them a second chance. So here they get to review the data again, review any additional feedback, and maybe try or adjust their conclusions as necessary. So here's an example of the review activities in this particular lesson. So we only focused on 3 particular pathways because these particular pathways are what are coming up in the experiments. So just to kind of get their brains focused a little bit on the content at hand. And so here we've used some figures from Dee's textbook, and then we've left some blanks for the students to drag and drop the correct hormones in the pathway. And again, these are low stakes and instant feedback, meaning that the students can then immediately check their answers, adjust their notes, and adjust their learning in these pathways as well. And so not only do the students see the correct, green check or maybe a red X if they mix something up, but there's also some text at the bottom of the feedback panel just to kind of provide them with some additional information. So really that's up to the instructor how much feedback they want to provide, but in the end, I personally feel, the more the better. After they've completed those review activities, this is where they get to apply some of that knowledge. And so this is where their students are filling in an endocrine prediction chart. So we've set the students up telling them that some exogenous hormones are going to be administered. And so they need to predict what's going to happen to the glands downstream of that pathway, or maybe these glands aren't in that pathway, but the students need to sort that out. And so with this, the students fill in the chart signifying whether the tissue increases or decreases as a result of these exogenous hormones. This also then, as a result, will ask the students to apply their knowledge of negative feedback loops to assist in filling out this chart as well. So again, applying some of that information too. And again, this was also a low stakes activity where the students can check their answers, receive feedback as they fill out the chart. And again, up to the instructor how much feedback they want to provide, but you can also add text in addition to the correct answers to help the students kind of determine how to fill out the chart and where maybe some misconceptions may lie. So after the review and the application practice as part of the lesson, we've now moved on to the experiment. So here students are led through a scenario which places them in the role of a researcher, right? So they get to assume the role of a scientific researcher, which the students often find is kind of fun. The students are tasked with using the presented data to diagnose the exogenous hormone that was administered to the rats. And so here there's 3 different experiments that the students will need to evaluate. And this data actually is what comes from the paper that I referenced earlier in the talk. And so there's actually 6 different experiments in that paper that you could choose from, or you could do all 6. We only focused on 3 for this particular activity. Each experiment itself has the students compare data from 4 different rats. And so each rat is presented, there'll be a little bit of text setting up the experiment, as well as kind of some descriptions there, as well as organ weights and body weights that the students have then to like synthesize and organize this information to then eventually diagnose or come up with a conclusion as to what the exogenous hormone was administered to result in these changes. And so one of the features that is particularly helpful in LT for this type of work is the notebook feature. And so I have that outlined there in the bottom in red. And so the notebook feature allows students to take notes in LT, and then anything they type in that notebook is going to be carried out throughout the lesson. So as they progress through the experiments, their notebook goes with them. So think of it kind of like a lab notebook. And I actually provide my students with a very detailed rubric in terms of what I want to see in their notebook, because I tell them that a lot of their grade is actually going to come from what's written in the notebook, because that's where their rationale is, right? So it's one thing to kind of take a guess as to what the hormone might be. It's another thing to tell me why you made that guess and where the rationale, how you're drawing your conclusions, what data are you using to come to that hypothesis. And the students also have the ability to revisit that hormone prediction table that they created previously, so using some information that they've already created to solve these little medical mysteries. As they get to the end of an experiment, they're going to be asked to submit an answer. And so this is where they don't get the same instant feedback, but they do have to commit to a specific answer. The students' notes are carried over as part of the notebook, and again, they get to use their notebook to justify their answer. And also help them make the decision. And so again, when they want to commit an answer, they now have this check answer option. You're being told, okay, this is it, right? You're now submitting your answer, and they can adjust their notebook accordingly as well. But again, we give them a second chance. So at the end, after they've completed all 3 experiments, and maybe they've received some feedback along the way. So I actually, because this was an asynchronous kind of a virtual option for me, I also encouraged them to visit a Zoom meeting with me and other students. And so before they completed their second chance, when they joined the Zoom meeting, they were able to talk to me and ask questions and see if they needed any additional information or get assistance. And then they could go to their second chance page, edit their notebook, make any adjustments, update their rationale and conclusions, and then take another chance at the hormone diagnoses as well. So again, this allows for a really great step in students' learning in terms of reflection and metacognitive practice, which is an excellent thing for them to learn how to do and to practice themselves. And again, the notebook is available for editing and review this entire time. At the end of the lab, a student report is generated, and this is what the instructor will assess or what I always look at. So it's automatically sent to me as a PDF, and it includes everything that they answered on the review questions, the experiment questions, the tables, and then their entire notebook. Because I told you the entire notebook is where I'm really looking at to assess their grade and their thought process. Again, if you wanted to set this up as a group report, you could do that as well, but I had them all individually sent to me too. One of the other things I like about LT is that I can get student feedback. So at the end of every lab, I always have like 1 or 2 questions where I just ask the students, what did you think? What worked? What didn't work? And it just kind of helps me when I prepare lessons in the future of what they liked and what they didn't like, or if I have specific questions, I can ask them as well. And that's all generated as part of the report too. Well, that was it, pretty short. I want to thank Dee for her help in constructing this lesson, as well as the American Physiological Society, because without them and the support from the Teaching Career Enhancement Award, actually, they funded LT for my courses. So my students got to work with me and teach me a lot as I learned LT as well. And then all the staff at 80 Instruments for their help setting up my LT lessons and getting my courses up and running. So thank you all.
Awesome. Thanks for that. I see you did the Donna approach and went right up to time. So I don't have time for questions at the moment, but potentially, yeah, if we have some time right at the end of the event, maybe we could come back to those questions that Ryan and Terry have asked. If not, then we can pass them on to you after the event and you can answer them offline. So
Okay.
cheers.
Yep, I just wanted to mention, Jennifer, lots of interest in your lessons. So there's a lot of chat going on around it. So yeah, very good to see.
Awesome. Thank you very much, Jennifer.
Awesome. Happy to share.